Hello, this is Mr. Buss, and in this video I'm going to show you how to do day 2 of the Taste of Genetics lab. This is the equipment that you'll need at your lab station. One pipette, I am going to use the smaller 2 to 20 microliter pipette, some pipette tips, waste container for the tips. You'll need your DNA sample that you extracted from your saliva, from your cheek cells on day 1. You'll also need, I have these prepared on my desk up front, the TAQ master mix. This is going to have the polymerase enzyme in it that's going to add nucleotides, and the primer mix. So the primer is going to contain the primers which are going to anneal to the DNA. So basically in this day 2, what we're going to do is we're going to take the DNA that we have in here, which is your entire genome, a small amount of DNA, but it contains your entire genome, and we're going to amplify only the region that the primers anneal to on either side of a small segment of the gene that contains the SNP, or single nucleotide polymorphism, that we're going to be looking for, so whether or not you're a taster. So we're going to make billions of copies of that small segment of DNA. For your protocol, you're going to be on page 23 of the booklet at your table. And so we're going to start with step 1, and we're going to end this video on page 24, step 7, so day 2 procedure. So the first step says to add 5 microliters of the primer mix and 10 microliters of the TAC PCR master mix to your DNA. So use a clean pipette tip every time. It's already set to 5, so I'm going to add the primer mix first, 5 microliters. So 5 microliters. Not very much. It's okay if it doesn't go all the way down, because you'll centrifuge this briefly. Discard the tip. And 10 microliters of the TAC. So rather than pipette 5 twice, I'm going to just adjust this to 10. So 100 isn't 100 because that's a decimal place 0, so this is 10.0. Okay, I've added my solutions to my DNA extract from yesterday. After I've capped the tube, I'm going to gently flick the tube, and that will allow the solutions to mix together, the primer, the enzyme, and the DNA sample. So now I'm going to do step 3. I'm going to centrifuge this for 15 seconds, 8,000 RPM. Okay, after that step, all the solutions should be at the bottom of the PCR tube, and we're going to place the sample in the PCR machine. Okay, once everyone's placed their sample in the PCR machine, we'll run the protocol. So we've done step 4. I'm going to set up the protocol now using the app. When it's done, we can remove the tube and we can store it in the fridge until tomorrow, or until day 3.